Nucleic acids were first discovered in the nuclei of cells, marking a pivotal moment in biological science. These large organic molecules are fundamental to life, containing carbon, hydrogen, oxygen, nitrogen, and phosphorus. Their discovery opened the door to understanding how genetic information is stored and transmitted. Nucleic acids come in two main varieties: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Both play crucial roles in the functioning of cells and the inheritance of traits. In this video, we'll explore the structure, function, and importance of these molecules, setting the stage for a deeper dive into the central dogma of genetics. Deoxyribonucleic acid, or DNA, is a macromolecule composed of two polynucleotide chains that twist together to form a double helix. This structure is the carrier of genetic information in all cells and many viruses. DNA's double helix is not just a beautiful shape, it's a functional design that allows for the storage and replication of vast amounts of information. Each strand contains sequences of chemical bases that encode the instructions for building and maintaining an organism. DNA is the foundation of heredity, ensuring that traits are passed from one generation to the next. Ribonucleic acid, or RNA, is the second type of nucleic acid. Unlike DNA, RNA is usually single-stranded and acts as a messenger, relaying instructions from genes to guide each cell's synthesis of proteins. RNA plays a vital role in translating genetic information into functional proteins, which are essential for countless cellular processes. Without RNA, the genetic code stored in DNA would remain unused, and cells would not be able to produce the proteins necessary for life. At the end of this lesson, you'll be able to describe the structure of DNA and RNA molecules, understand how information is stored within these molecules, explain the rules of base pairing, and grasp the central dogma of protein synthesis. You'll also learn how the genetic code and nucleotide sequences prescribe the amino acid and protein sequence, connecting the molecular world to the traits we observe in living organisms. In the early 1950s, researchers sought to understand the detailed molecular structure of DNA. The breakthrough came in 1953 when James Watson and Francis Crick proposed the first correct three-dimensional structure. DNA consists of two long chains of subunits twisted around each other, forming a double-stranded helix. This right-handed helix is composed of nucleotides, each made up of a nitrogenous base, a pentose sugar, and a phosphate group. The arrangement of these components is key to DNA's function as the genetic blueprint. DNA's nucleotides feature four nitrogenous bases: adenine A, thymine T, cytosine C, and guanine G. Adenine and guanine are purines, larger double-ring bases, while thymine and cytosine are pyrimidines, smaller single-ring bases. The base pairing between A and T and between G and C is known as complementary base pairing. This pairing ensures the accuracy of genetic information during replication and allows DNA to code for the vast diversity of life. Each DNA nucleotide contains a 5-carbon sugar called deoxyribose, which attaches to the nitrogenous base. The phosphate group alternates with the pentose sugar to form the backbone of the DNA strand. This backbone provides structural stability and directionality, allowing DNA to maintain its double helix shape and function effectively as a carrier of genetic information. DNA strands have polarity, determined by the direction in which nucleotides are pointing. In double-stranded DNA, the paired strands are oriented in opposite directions, known as antiparallel. The 5 end of one strand aligns with the 3 end of the other, ensuring proper base pairing and replication. This antiparallel arrangement is fundamental to DNA's function and replication. Genetic material exhibits 4 key characteristics: replication, storage of information, expression of information, and variation by mutation. Replication allows DNA to faithfully copy itself, ensuring genetic continuity. Storage of information enables DNA to encode traits. Expression of information involves the flow of genetic instructions, and variation by mutation introduces diversity, driving evolution. There are 3 models for DNA replication: conservative, dispersive, and semi-conservative. The conservative model keeps parental strands together, the dispersive model mixes old and new DNA, and the semi-conservative model results in each new DNA molecule containing one old and one new strand. The semi-conservative model is the general mechanism by which DNA duplicates, ensuring accuracy and continuity. DNA synthesis involves several enzymes called DNA polymerases. Polymerase I removes primers and fills gaps. Polymerase II initiates and elongates DNA strands. Polymerase II holoenzyme elongates chains and proofreads. And polymerases IV and V repair damaged DNA. These enzymes ensure the fidelity and repair of DNA during replication, maintaining genetic integrity. Replication begins with the breaking of hydrogen bonds between bases of the 2 antiparallel strands. Helicase splits the strands at the origin of replication, creating a structure called the replication fork. This fork is the site where new DNA strands are synthesized, ensuring accurate duplication of genetic material. The initiation point of the 3-5 parent chain, RNA primase binds and attracts RNA nucleotides. These nucleotides form hydrogen bonds with DNA bases, preparing the strand for elongation. This step is crucial for starting DNA synthesis, especially on the lagging strand. DNA replication differs for the leading and lagging strands. The leading strand is synthesized continuously, while the lagging strand is synthesized in fragments called Okazaki fragments. DNA polymerase reads the template and adds nucleotides, ensuring both strands are accurately copied. On the lagging strand, DNA polymerase I reads fragments and removes RNA primers. DNA polymerase and DNA ligase then close the gaps, ensuring each new double helix contains one old and one new chain. This process maintains genetic continuity and accuracy. Genes serve as the repository of all hereditary characteristics in organisms. DNA's ability to encode a nearly infinite variety of gene products is essential for the diversity of life. The storage of information in DNA allows organisms to inherit traits and adapt to their environments. The expression of genetic information involves transcription and translation. Transcription produces mRNA, tRNA, and RNA, while translation uses these molecules to build proteins. This flow of information is the basis for cellular function and the manifestation of traits. RNA is a complex compound of high molecular weight that functions in cellular protein synthesis. It replaces DNA as the carrier of genetic codes in some viruses. RNA consists of ribose nucleotides attached by phosphodiester bonds, forming strands of varying lengths. The nitrogenous bases in RNA are adenine, guanine, cytosine, and uracil, which replaces thymine found in DNA. Ribose sugar in RNA contains a chemically reactive hydroxyl group, making RNA prone to hydrolysis. This chemical ability is one reason why DNA, which lacks this reactive group, evolved to be the preferred carrier of genetic information in most organisms. RNA's structure was first described by R.W. Holley in 1965. Transcription is the process by which RNA is synthesized from DNA. It involves three essential steps: initiation, elongation, and termination. RNA polymerase attaches to a specific DNA sequence called the promoter, aided by the sigma factor. Once bound, the sigma factor dissociates, allowing transcription to proceed. During elongation, RNA polymerase moves along the DNA strand, separating the two strands and complementing DNA nucleotides with RNA nucleotides. Thymine is replaced with uracil, so adenine pairs with uracil. As the segment is transcribed, the DNA strands reanneal, and a single-stranded RNA is produced. Termination occurs when another protein, the terminator factor Rho, binds to the enzyme and dissociates it from the DNA. This prevents further transcription and produces RNA complementary to the anticoding strand. The resulting RNA can be mRNA, tRNA, or RNA, all essential for translation. mRNA provides the template that gives the code for the amino acid sequence of proteins. Every 3 nucleotides of RNA, called codons, specify one amino acid. This triplet code is fundamental to translating genetic information into functional proteins. tRNA is necessary to decipher the genetic information coded within mRNA. Each carries the amino acid that corresponds to its anticodon. During translation, the anticodon in tRNA binds to the codon in mRNA, ensuring the correct amino acid is added to the growing protein chain. RNA complexes with proteins to form ribosomes, the site of protein synthesis. Ribosomes consist of small and large subunits, which join during protein synthesis to form a complex with 2 important sites, the A site acceptor and the P site peptidyl binding. These sites facilitate the translation of mRNA into proteins. Translation begins with the activation of amino acids. Each amino acid is attached to its respective tRNA, a process requiring energy from ATP and mediated by aminoacyl synthetase enzymes. This ensures that each carries the correct amino acid for protein synthesis. Initiation codon AUG is always oriented on the 5' end of the mRNA. Initial steps involve the attachment of the first R-tRNA complex with formulated methionine to the 30S ribosomal subunit. Initiator factors and GTP energy facilitate the assembly of the ribosome and the start of translation. Once the initial amino acid is set at the P site, a new R-tRNA complex enters the A site, complementary to the next codon. Peptidyl transferase catalyzes a peptide bond between the amino acids, and the ribosome moves along the mRNA, repeating the cycle until the entire protein is synthesized. Translation ends when the ribosome encounters one of 3 termination codons, UAA, UAG, or UGA. These codons do not code for any amino acid, but signal the release factors to dissociate the polypeptide and tRNA from the ribosome, completing protein synthesis. The genetic code is the set of rules by which information encoded in DNA or RNA sequences is translated into proteins. Genes that code for proteins are composed of trinucleotide units called codons, each specifying a single amino acid. This code is universal, used by almost all living organisms. The genetic code is written in linear form using ribonucleotide bases as letters. Each codon contains 3 ribonucleotides, specifying one amino acid. The code is unambiguous, degenerate, contains start and stop signals, is commaless, non-overlapping, and universal. Let's walk through an example. The antisense strand of DNA is given, and from it, the sense strand is determined. The mRNA is then transcribed from the sense strand, and finally, the polypeptide chain is translated. Each codon in the mRNA corresponds to a specific amino acid, demonstrating the flow of genetic information from DNA to protein. DNA contains the sugar deoxyribose, while RNA contains ribose. The only difference is that ribose has one more hydroxyl group than deoxyribose, which has a hydrogen attached to the second carbon. This small change affects the stability and function of each molecule. DNA is a double-stranded molecule, while RNA is single-stranded. This structural difference influences their functions. DNA is stable under alkaline conditions, while RNA is not. DNA stores and transfers genetic information, while RNA directly codes for amino acids and acts as a messenger. DNA uses the bases adenine, thymine, cytosine, and guanine, while RNA uses adenine, uracil, cytosine, and guanine. Uracil replaces thymine in RNA and lacks a methyl group. This difference is crucial for the processes of transcription and translation. Central dogma describes the flow of genetic information. DNA is transcribed into RNA, which is then translated into proteins. This process is fundamental to all living organisms, connecting the molecular world to the traits we observe. Complementary base pairing ensures the accuracy of DNA replication and transcription. Adenine pairs with thymine in DNA and with uracil in RNA. Guanine pairs with cytosine in both. This pairing is the foundation for genetic fidelity and information transfer. A genetic code is degenerate, meaning that a given amino acid can be specified by more than one triplet codon. This redundancy provides a buffer against mutations and ensures the robustness of protein synthesis. The genetic code contains start and stop signals. The start codon, AUG, initiates translation, while stop codons, UAA, UGA, UAG, terminate it. These signals ensure proteins are synthesized accurately and efficiently. With only minor exceptions, the genetic code is universal. A single coding dictionary is used by almost all viruses, prokaryotes, archaea, and eukaryotes. This universality underscores the shared evolutionary history of life. DNA replication is a highly accurate process. Thanks to the proofreading abilities of DNA polymerase, errors are corrected, ensuring the faithful transmission of genetic information from one generation to the next. Mutations introduce variation into the genetic code. While most mutations are neutral or harmful, some drive evolution by creating new traits. This variation is essential for the adaptability and survival of species. After translation, the polypeptide chain may undergo further processing to become a functional protein. This includes folding, modification, and transport, ensuring proteins perform their specific roles in the cell. DNA and RNA perform distinct functions in humans. DNA stores and transfers genetic information, while RNA codes for amino acids and acts as a messenger between DNA and ribosomes. Together, they ensure the continuity and expression of life. In summary, nucleic acids are the foundation of genetic information. DNA and RNA work together to store, transmit, and express the instructions for life. Understanding their structure and function is essential for grasping the principles of genetics, evolution, and cellular biology. These molecules connect the microscopic world of molecules to the macroscopic traits we observe in living organisms, highlighting the unity and diversity of life.